Welcome back once again to Introduction to Neuroscience, where the topic for the next several lectures will be visual perception and vision, the neurobiology of how it is that our nervous system can detect light, as well as process those visual information into a form that allows us to make sense of the visual world around us. In talking about vision, we're going to start at the beginning by talking about the retina. And the retina is, well, it's at the back of your eyeball, so we're going to talk about the anatomy of the retina in a second. But I wanted to give you kind of a bigger view of the visual system to begin with. So your eyeballs are, I have two of them, you're looking at them right now. Those eyeballs are going to be attached to the rest of your nervous system by the optic nerves. This here is the ventral view of a human brain. So kind of like, if you opened up my brain and looked at it from underneath. This is the ventral view. And what you're seeing here are parts of the optic nerves. So the eyeballs would actually be, left and right up here on the top here. And the two of them will be attached to these big, thick cables, because there's going to be lots of wires that are connecting your eyeball to the rest of your nervous system. And they're going to actually cross over at this point right here, this little X here, which is called the optic chiasm. The reason they cross over is because the left side of your visual field actually goes to the right half of your brain, and the right side of your visual field goes to the left half of your brain. If you think about what your eyeballs are seeing, because most of our eyeballs are facing forward, each of your eyeballs sees half of the left half of your brain visual scene and half of the right half of the visual scene. And so the cables actually have to cross over in this way because the left half of your left eye goes to the right side, but the right half of your left eye goes to the left side. Okay, so that's why there's this crossing here between the two different eyeballs. There's a diagrammatic view of the same thing here where you can see the retina, which is the piece of tissue, the piece of neural tissue at the very back of your eyeballs that has the visual fields. They cross over at the optic chiasm where the left visual field goes to the right half of the brain, and the right half of your visual field goes to the left half of the brain. And so they have this path here where they start to diverge and then connect to the part of the thalamus called the lateral geniculate body, the LGN. This then, of course, then connects to the rest of your nervous system via the visual cortex, which is the part of cortex that's in the very back of your head in the occipital lobe. Now, I know that was just a lot of words, but I wanted to give you a lay of the land and kind of just give you a map of where we're headed. The rest of this lecture, we're mostly going to be concentrated on the eyeballs and the retina, but this is sort of where the rest of the lectures will go in the next several videos. So this here, we're going to summarize this pathway by this rather simplified diagram. Now, I show this diagram not because it's actually correct, but because this is kind of a mental framework on which we're going to hang our knowledge of how visual computation works. Photodetector, detection of actual photons and light, starts at the retina, which passes this information into a part of the midbrain called the thalamus. In particular, it's a part of the thalamus called the LGN, the lateral geniculate body, the LGN. We think of the thalamus somewhat as a relay station, so kind of an intermediary for sensory information in between wherever they're actually detected and where they're going in the different cortical areas. This is a generic feature of lots of different sensory systems, not just vision. And so you'll see this diagram throughout the rest of the lectures as well when we're talking about the other senses like auditory sensation and tactile sensation, etc. Okay, so retina goes to LGN, LGN then goes to cortex. In particular, for vision, it goes to the part of cortex called V1, V1 for primary visual cortex. From V1, it gets passed on to higher order visual areas, and they have fancy names like V2 and V3 and V4 and so on and so forth. You can kind of imagine the rest. They also have other fancier names, but that's a topic for a later video. For now, though, I do want to preview one thing, which is a theme that we're going to be coming back to in the rest of the lectures in this series on sensing and action, which is that this vastly simplified diagram is actually pretty wrong. Okay, I'm going to point out a couple of different ways in which we know it to be wrong. The retina does project to the LGN. That part is correct. The LGN does project to the V1 in cortex. That part is also correct. But what we were ignoring in the previous diagram is the fact that V1 actually projects back to the LGN more than the LGN projects to V1. Now, as you can see here, these feedback loops is an essential feature of not only the visual system, but of every other system, including sensory motor systems in your entire nervous system. And so I'm going to persist in talking about these visual pathways of communication in this overly simplistic way, but I did want you to keep in mind that we already know that to be wrong, okay? But this is kind of a mental framework for thinking about how information is processed throughout the nervous system. And we're going to be talking about first the retina, which is the part of which is where it all starts, right? So the photodetection has to start at the retina. So in the beginning there was a sun, and the sun emitted photons. Okay, so that we know that to be true. And we needed some kind of hardware apparatus, some kind of biological piece of machinery that's able to detect photons from the light. But let's talk first about what light actually is. As you remember, probably from physics class, light is electromagnetic radiation. And electromagnetic radiation actually comes in a variety of different wavelengths that are vastly larger than all of the different wavelengths of photons that we can actually perceive as humans. The visual spectrum occupies this really thin region of all the possible different types of light that possibly exist. We can now build engineered detectors to detect all of these different types of light and also produce them. But humans, with their own eyes, can only see this part of the visual spectrum with wavelengths that are approximately between 380 and 750 nanometers. Now, as you can see from this diagram here, wavelength, as it increases, an equivalent way of describing that is saying that the frequency decreases. So increasing frequencies means that light gets longer and longer a wavelength, and longer wavelength is the same as lower frequency. Okay, those are exactly the same thing. It's just one divided by the other. One divided by frequency gives you wavelength. One divided by wavelength gives you frequency. So I kind of talk about those interchangeably. The other thing that's that's important to remember here is that lower wavelength of light and lower wavelength of light and higher frequencies of light also correspond to higher energy. Okay, so when I say lower wavelength this way, it also means increased frequency, which also means higher energy. Those are kind of equivalent concepts in the context of waves, including light, but also of other waves as well. So that's light, right? There's these photons flying around, but it's not obvious how we can turn that into electrical potential. So if you remember in the series of lectures we had in the beginning of the course on electrical potentials and how cells maintain the electrochemical gradients and are electrically activatable, we talked about activating neurons in terms of depolarizations and hyperpolarizations. And these events kind of just happen. They just kind of happen by themselves. We didn't really talk about how, right? A cell could be depolarized because something else was already depolarized. Now we're sort of at the beginning of that process in the context of vision, how it is that we can actually trigger a cell to become in a different excitable state when an external stimulus from the external world activates or deactivates that cell. So the nature here of the thing that's doing the activating is light. It's a photon. And as we talked about here, photons carry energy. So we're going to use that as a clue of how we can activate cells in the visual system. So the retina is the very last back part of your brain, of your eyeball. So here's a human eyeball. You can see it has, well, first of all, it's beautiful to look at, but it also has the central portion that's black, appears black in this picture, which is the pupil. So light goes through the pupil, which is kind of like an aperture. Okay, and it goes to the very back of the eyeball into a piece of photoactivatable tissue called the retina. So for this reason, a lot of people have made the analogy that your eyeball, and all the lenses that go with it, plus your retina is kind of like a camera, right? So you have a shutter, which is your eyelids. You have an aperture, which is kind of like your pupils, right? And then there's a CCD, a light activatable piece of machinery that's photosensitive at the very back, which is your retina, and that's something like a CCD in a camera. And so this equivalency and this analogy is not exactly untrue, but what I hope to convince you in the next several lectures is that, in fact, the eye plus the retina is significantly better than what you can have in a camera. You can see this just by inspection. Like you can look at sometimes. Have you ever looked at something? And it's like, oh, this is amazing. This is a beautiful scene here. Let me take a picture of it. You bring out your camera, you take a picture of it, and you're disappointed by the result. Well, this is what I'm talking about here. We're going to be talking about the neurobiological basis of what your eye and your retina and your visual system do that is so much more than what can possibly be achieved with modern shutters and apertures and CCD in camera technology. And we're talking about how that actually happens. Okay, so here's your eyeball. And now we're kind of looking at a slice of it, a vertical slice of it, where we're looking at the lens, which is on top. Okay, I'm not talking about the physics of the lens very much because we're going to be focusing mostly on the neurobiological basis of vision. And so just assume that there's a lens up there that does actually focus light in some way. The light then comes down through this viscous water-filled region of the eyeball, okay? And it hits the very back of the eyeball where the retina lives. So this is a piece of tissue that contains all of the photoreceptors of your nervous system that contribute to image-forming vision, okay? So if you zoom in to a cross-section of the retina now, you see that it's not only full of photoreceptors. It's got a couple of other different types of cells too. In fact, the photoreceptors themselves are actually in the very back of the retina. It's in the very bottom layer. So I'm going to tell you about the other layers of the retinas in a second, but let's just think about this for a second. The very back of the retina has the photoreceptors, which has two consequences. Okay? The first consequence is that light actually has to go through all of the other layers of cells before it hits a layer of cells that's actually responsible for actually absorbing those photons and detecting light. Kind of a funny thing that happens. If we zoom in further and look at the different cell types here, there's actually a lot of different cell types. Actually going into the computations of what those different cell types do is a subject of the subsequent lecture. But for now, I'm going to tell you about 3 large groups of cells that are in the retina. We've already talked about the photoreceptor cells. These are the cells that are photosensitive. We're going to start talking about how they detect light. These cells are connected to the bipolar cells, which help separate the different channels of light that we are detecting into more informative, more interpretable pieces of information. This information is then gathered by ganglion cells, or retinal ganglion cells, and these accomplish some additional processing, and they're also the only ones that actually pass messages to the brain. So none of these other cells have any outputs to the rest of the brain. Only the retinal ganglion cells have outputs to the rest of the brain. So the axons from the retinal ganglion cells then go up, and then they go to the optic nerve, which we saw in the previous picture. The optic nerves then converge from the 2 eyeballs into the optic chiasm. They split and do this thing, which then they keep going into their respective parts of the LGN. Okay, so that's kind of where that information is going. In a human, we have approximately 1 million axons from different retinal ganglion cells. So that's approximately the number of channels of information that you can actually have from one eyeball in a human. Light, as I talked about earlier, actually goes through all of those layers of processing, those extra cells, before they hit the photoreceptor cells. So that has a couple of different consequences. So let's talk about the photoreceptor cells for a second. These are the primary sensory neurons that are responsible for detecting light. And in humans, we have 2 types of photoreceptors. We have our rods and our cones. They're named because one of them looks like rods and the other one looks like cones. You're going to see an electron micrograph of them on the right-hand side there. The rods tend to be much larger and the cones are a little bit smaller, but they're fundamentally detecting light in similar ways at the molecular level. So we're going to be talking about that in the next couple of slides. So the things that makes rods and cones different is that rods are not really color selective. They're super fast and they're sensitive to very few photons hitting them. Cones, on the other hand, do have color selectivity, but they're much slower and they're only activated with way more photons. Okay, so color vision is something that we have in highlight situations, and in low light situations, most of your vision is then reduced down to this more monochromatic, only light sensitive part, because it's mediated by cones in highlight and mediated by rods in low light situations. Now, I talked about the fact that your photons actually have to go through all of the retinal ganglion cells and all of the bipolar cells and all of the other cell types in your retina before it hits the photoreceptors. So kind of, an interesting design choice, mostly because it wasn't designed that way. And one of the other consequences of it is that because the retinal ganglion cells have to send axons to the rest of your brain, the axons have to go through in front of all of the photodetectors, and then they have to punch through the photodetectors so that they can actually get to the rest of your head. Okay? So that is why we have this bundle of optic nerves that has to interrupt the line of photoreceptors at the very back of your retina, giving us the blind spot. So there's a spot in the middle of your left eyeball and your right eyeball, a little off center, respectively, where you don't have photoreceptors. So you're actually blind to that region. You can't see that region of your visual scene. The reason this doesn't bother us most of the time is that, well, there's two reasons. One, you have two eyes, and so your blind spots in the left eye and the right eye are not in the same spot, and so your brain kind of fills that in. But even if you close one eye and look at it, it's not terribly obvious where the blind spot is. I don't really notice it. And that, again, is an effect of higher visual processing. Our brains are really good at filling in things as if it were there, even though you can't actually see it. But you can kind of see it if you close one eye and you very carefully hold something that is away from you by a certain amount, and you'll eventually be able to find that there's a spot where you can't actually see that small object, and that is where your blind spot happens to be. You don't really notice it most of the time, but I assure you it most certainly is there. This choice, it's not really a choice, this idea that there's a blind spot in our eye because of the way that our photodetectors are inverted, where the photosensitive neurons are at the very bottom, at the very back of your retina, is not necessarily true of all animals. An octopus eye, for example, has the opposite configuration, where the photoreceptors are on the outside, like facing the lens, and then the retinal ganglion cells are in deeper layers. And so for octopus eyes, they don't have this issue. Octopus eyes do not have a blind spot because their retina, the cell layers of the retina is actually inverted. So that's really cool, but there's also other cool animal visual cells that work really differently than ours. I won't go into a lot of the details here, but I did want to acknowledge here that just because we see in a certain way doesn't mean it is the only way to see, and is by no means the best way to see. So some examples of the diversity of visual systems I'm showing here are there's insect eyes that don't work like ours. They don't have a single lens and a retina in the back. They have what's called a compound eye, where each of those little dots is actually a little mini eye with ommatidia. Okay, so they kind of focus light individually. They have kind of a pixelated vision just like we do, but they accomplish it using an entirely different mechanism. Here are pictures of a scallop eye. Okay, so scallops have eyes, and their eyes are actually really cool as well. They have hundreds of them that are kind of on the outside of their scalloped edges. And these eyes have lenses. They have prototypical lenses that actually do focus light, and people believe that it's possible that they have some image forming capabilities as well. Down here we have a mantis shrimp, and the mantis shrimp have eyes that are able to see way more colors than we do, and that's something that's really puzzling why they would do so. They also see something called polarized light. So if you have a pair of sunglasses, I don't know if you've ever bought a pair of sunglasses that say polarized on them, and if you take two polarized lenses, right, you see that if you put them parallel to each other, everything's fine. If you start turning one of them so that they're orthogonal to each other, they actually block all light together. And it's because light from the sun actually has polarization, and it's something that our eyes don't see. The reason you don't see it is because your eyes literally can't detect polarization in light. Well, not true of mantis shrimp, and lots of other invertebrate species have this capability of seeing polarized light. We think it has something to do with being able to figure out where the horizon is when they're in the ocean and trying to hunt, for example. But that's a capability that they have that we also don't have in our visual system. Okay, so that's all really cool. I can give you a whole lecture on that. But for the moment, let's go back to the human eye, okay? So the human eye, I told you that has photoreceptors. And so here we're going to be digging a little bit more into the photoreceptors and thinking about how these photoreceptors actually work. Here are pictures of a couple of rod cells. And the way the rod cells work is that they're packed full of these rhodopsins, these photosensitive proteins that detect photons. And in order for these rods to be really sensitive to photons that are incident on the retina, you want to pack these photosensitive rhodopsins, as many as possible, really tightly packed. And so, not surprisingly, the photoreceptor solution to this is to basically stack the membranes into this like really Swiss pancakey shape, where there's tons of membranes that are stacked really, really close together, and there's a ton of them, increasing the effective surface area of this membrane by a huge margin. And all of those membranes, these rhodopsin discs, are just chocked full of rhodopsin proteins. Now, these rhodopsin proteins are G-coupled protein receptors, GPCRs. If that sounds familiar to you, it should, because as I told you before in the lecture on metabotropic postsynaptic receptors, GPCRs are everywhere. They are some of the most useful and versatile family of proteins that we know of, not only in the nervous system, but in the rest of the brain as well. And so go back to that lecture if you want to review on what GPCRs are. So the short version is that GPCRs are a family of proteins that are super adaptable, and they're transmembrane proteins, so they have to be bound in membranes. And that's why these photoreceptors have to have pancake layers of membrane so that you can pack these membrane-bound photoreceptors in them, okay? All right, cool. So that's rhodopsins. Okay, now they're GPCRs, they're G protein coupled receptors, so you already know that there's some kind of conformational change that's going to happen when a light hits it. So in the case of metabotropic receptors, we already talked about how in response to being bound by a neurotransmitter like glutamate or whatever, they will have undergo some kind of conformational change. In the case of rhodopsin, that conformational change is going to be triggered not by the binding of some kind of small chemical, but by a photon hitting it. But proteins are not photosensitive by themselves, usually. So there has to be some kind of photoactivatable molecule that is working in concert with the rhodopsin to get this to happen. In the case of rhodopsin, that molecule is retinol. Now, retinol is that small molecule you see down there, and it can take on one of two different conformations. In the absence of light, retinol comes in 11 cis conformation, and that's what's bound to the rhodopsin when there's no photon. Upon hitting it with a photon, the energy for the photon gets absorbed by the retinol, the cis retinol molecule, and causes it to change conformations into a trans conformation of retinol. That is the molecular, that's actually the biochemical basis of photoreception. Fundamentally, it's a photon hitting not the GPCR itself, but a small molecule of retinol that's bound to the GPCR that causes this transformational change. The cis retinol becomes a trans retinol. That causes the conformation change in the rhodopsin protein, and it's that signal that cascades down through a series of signal transduction, and that causes the photoreceptor to then respond, oh, there was a photon, I saw a photon. So the retinol pigment has a kind of an interesting story here. You may have heard at some point somebody tell you, oh, eat your carrots, it's good for your eyes. Now, why do we think eating carrots is good for your eyes? So the story here is that retinol in your body is metabolized from beta carotene. And beta carotene is this molecule that's found in abundance in carrots. Carrots are not the only ones that have it. You can get it from other different types of dietary foods that you can ingest. But the bottom line is, however you get it, when you have beta carotene, it becomes metabolized by your body and made into 11-cis retinol, and you need to make 11-cis retinol to put it into your rhodopsin so that you can actually detect light. Now, is more carrots and more beta carotene actually better? Up to a point, right? If you don't have any of it, then you won't be able to make retinol, and then you won't be able to see. Most of us have plenty of beta carotene in our normal diet, and so eating even more carrots does not make your vision even better, even though this is the molecule that we need in order to be able to see. So we have our GPCR, we have our rhodopsin. It has a retinol that's bound to it, so the conformational change triggers a change in the rhodopsin protein. And the rhodopsin, when it's activated, so it goes from the dark configuration, light hits it, the cis retinol becomes trans retinol. This activates the GPCR, which is the rhodopsin. The GPCR then activates the alpha subunit of the G protein. And what ends up happening is something that's slightly counterintuitive. Okay, we'll talk about why in a second, but I'm just going to tell you the fact of what happens. Photoreceptors are a type of neurons that don't spike. They do not have action potentials. Not only do they not spike, they have these graded potentials, so they only are even more or less depolarized. They don't depolarize in response to light, they actually hyperpolarize in response to light. The way that works is that in the absence of light, there is a series of calcium and potassium ion channels that are open. So when there's no light, there's actually a depolarization, a steady state depolarization of these photoreceptors. You can see that because the resting potential of these photoreceptors is actually at -30 millivolts. If you review the videos earlier about the resting potential of neurons, you'll remember by our derivation with the Nernst potential and the resting potential of the neurons that a typical neuron is closer to something like -6 60 millivolts. And so photoreceptors is already substantially more depolarized at rest in the absence of light than a normal neuron that we talked about, generic neuron, had been. So you have a neuron that is normally a little more depolarized than the generic neuron. When the light hits the rhodopsin, what happens is that instead of opening a cation channel, it actually closes the cation channel. It closes that channel, stopping the flow of calcium and sodium into the cell. Kind of counterintuitive. I'll say it again. Photoreceptors do not have spikes. They only get more or less depolarized. And the way they signal that a light has been detected is actually by hyperpolarizing themselves rather than depolarizing themselves. Now, the amount to which they hypopolarize is going to be proportional to the magnitude of the photons, so the magnitude of light, how many photons have hit that particular photoreceptors. So if only a few photons have hit it, it's going to hyperpolarize a little bit less. If a bunch of light just hit it, then it's going to hyperpolarize a whole lot. That's how that works, okay? Now, these photons, as they hit it, are going to cause the photoreceptor to the photoreceptors are glutaminergic neurons, and so they have a glutamate synapses where they are going to release glutamate as neurotransmitters onto the next layer of cells. But first, let's talk a little bit about how it is that with this infrastructure that we just talked about, how we can actually see relatively well across 8 orders of magnitude in luminance in different conditions. Now, in sunlight, we have about 10 to the 5th luminance measured in terms of candelas per square meter. We can see in sunlight, we can see indoor light, we can see about the light of a smartphone display. You can kind of read at night if you care to. Don't do it, but we all do. You can see in moonlight, and you can also see in starlight. If you go out at new moon in the middle of a desert, and there's only stars in the sky, you can actually see relatively well. You know, you notice it is dark, but you can see more than not at all. And this is 8 orders of magnitude difference in luminance. And in terms of performance of a CCD type apparatus, this is absolutely astonishing in terms of our ability to flexibly and without really thinking that hard about it, see quite well across 8 magnitude of luminance in lighting conditions. How is this possible? So it turns out that the mechanisms for how we can see so well across 8 orders of magnitude are fourfold. The first thing that happens, and this is the fastest thing that happens, is pupil dilation. In low light conditions, your aperture opens up wide, and so you just literally have more photons going into your retina. That causes a 10 times ability to adjust to different lighting conditions. At lower light conditions, you actually switch from cone-based vision into rod-based vision. I've already told you that cones are less sensitive to light, rods are more sensitive. And so switching between cones and rods gets you about another 1000x in terms of how much light you can actually see. Over an even longer time scale, the photopigments in your retina actually adjust, and that gets you another 100 times difference in luminance that you can account for. So this happens over the course of approximately 30 minutes. So 30 minutes is about how long it takes the human eye to fully adjust to low light conditions. Think about that the next time you go stargazing, if you want to go out and see a meteor shower or something. If you spend even 1 minute turning the light on in your car or like reading news in your phone or something, you have to spend another 30 minutes to fully adapt to the dark conditions again. So just don't do it. That's how long it takes, okay? So the pupil dilation's super fast, right? Less than a second. Switching from rods and cones takes a couple of minutes for your light to adjust for that long. Over the course of the next 10, 20 minutes or so, what's happening in all of your photoreceptors is that in response to this chronic dark condition, your photoreceptors will actually manufacture and make more rhodopsin freshly loaded up with cis-retinal and loaded up into your photoreceptors so they're ready to detect very, very small numbers of photons. That whole process takes about 30 minutes. And so, you know, that's kind of a practical tip next time you go stargazing. Okay, so the human eye, like I told you, has rods and cones, but they are distributed not at all evenly in the back of your retina. The cones are actually found in the very center of your retina in this area called the fovea. That's the name for the very, very middle of your retina. And it's so thin that it's actually only accounts for about the center 1 degree or so. That's the only place you really have any concentration of cones. That's where all of your color vision lives. the density of cones outside of the fovea decreases rapidly. To compensate for all of that real estate taken up with cone cells, there's not really that many rod cells in your retina. So in the very center of your retina, you don't actually have that much light sensitivity. In fact, the peak of your rod photoreceptors is a few degrees away from the center. Okay, so that's actually your peak light sensitivity when things are relatively dark. The other interesting thing here is if you count the total number of photoreceptors, the human retina has way more rod cells than it has cone cells. Cones are only found in the middle. They're densely packed. There's tons of them, but they're not very light sensitive. And the rest of your retina is actually tiled by a variety of different cones. And so your peripheral vision is largely mediated by rods instead of cones. So that's what it looks like in a human. That slightly dark spot in the middle is the fovea, where we have a lot of acuity because we have tons of closely packed cones, so that we can kind of see very small differences in the visual scene. Not all animals have a fovea. There's lots of animals that just don't have this dense region in the middle or anywhere altogether that has a lot of acuity, and so lots of animals just don't have nearly the acuity that we humans and some other primates do. There's also animals where their fovea is not so much a spot as a line. And so a turtle retina, for example, looks like the following, where there is a line of photoreceptors that are really closely packed. And so for them, it's not a spot fovea, it's actually called a visual streak. Okay? So it's a line with closely packed photoreceptors, so their acuity is really high, even in some of the peripheral regions. They don't only have high visual acuity in the middle, they also have pretty high visual acuity in the sides as well. And that's something that is necessary for their ecology and to survive in the ecological niches that they survive in, unlike humans and other primates to whom we are most closely related. Rods are so sensitive to light that they have been demonstrated to be able to respond to single photons. Under fully dark adaptive situations, there are experiments that have been done, both psychophysically as well as in terms of single photon absorption. Here you're seeing the response of one rod photoreceptor in response to single photons being absorbed. And so that's sort of the peak of the performance of these rod photoreceptors. In fully dark adaptive situations, if you just wait around a really long time and for one single photon to hit one photoreceptor in your eye, it is in theory possible for a rod to detect a single photon. Kind of remarkable if you think about how difficult it is to build even a engineered apparatus to detect single photons. Another consequence of this is another practical tip the next time you go rock climbing out at night, as one does, and you find that you can't quite see right in front of you, right? The trick is actually you look slightly away from it. Right? You put the thing you're trying to look at not in the middle of your eye where your fovea is because you have no rods there. The peak fovea is a few degrees away from the center, and so if you are trying to look at it and you can't see it because there's not enough photons, look slightly away from it and you actually have a better chance of being able to resolve it because that's where the peak concentration of rod receptors in your retina are. Okay, so those are all the things that I thought to highlight about photoreceptors in the retina. To review, here are some key concepts. The first is that we have photoreceptors in your retina. These are specialized sensory neurons that are able to detect photons. And they detect so by using two different types of photoreceptors. We have the rods and the cones. The rods are really fast and they're really sensitive, but they're not really color selective. For a closer explanation of why they're not color selective and what color selectivity is, we're going to talk about all about that in the very next lecture. Cones are slower and fewer, but they do have color selectivity. And the human retina has way more rods than cones, and rods are mostly found away from the fovea in the peripheral regions of your brain, of your retina. Photoreceptors are able to detect light by a molecule called rhodopsin, which is a light-activated GPCR. Light-activated GPCR relies on a small molecule called a retinol, whose conformation from a cis configuration to a trans configuration in response to absorption by a photon is the thing that actually causes rhodopsin, it's a GPCR, to change conformations, and that's the first step in the phototransduction pathway in order for you to then perceive light. Photoreceptors, interestingly, do not spike. They have no action potentials. They have graded potentials, and they're actually hyperpolarized by a photon rather than being depolarized like we sometimes generically think of neurons are. This is not true the next stage of retinal transduction, and that's going to be the focus of the next lecture as well.